Okay, everyone. Today we are going to talk a little bit about PCR. So PCR, that is the polymerase chain reaction, and we're going to learn a little bit about how this works. So, and there are many reasons why a scientist or a geneticist would want to use this technique. So, for example, let's say we have a cell from any organism and we're interested in a gene from this organism. So here's the cell, and here is the nucleus here, and in here we're going to have our chromosomes, and somewhere in that chromosome we have the gene that we're interested in. And so I will zoom in on that gene, and let's say this gene is about 3,000 base pairs long. It's about 3,000, and that's going to include the promoter region, the transcriptional start site, the coding region, all these parts of a gene that we've talked about over the course of the semester. And then we'll have the terminator region with the terminator site. And what we want to do, and here obviously this isn't to scale, but I'm just showing the hydrogen bonds between these two strands of DNA. And what we want to do, let's say we want to copy this gene. So let's say we want to transfer this gene to another organism. Let's say a mouse, or how about a plant? For instance, we have a plant here, and this is a important plant, and we can sell it for a lot of money, but let's say the flower petals aren't very pretty. And maybe if we make the plant prettier, we can sell a lot more of this plant. So we're going to make a transgenic plant. It's going to make these flower petals really pretty. We have an idea that this gene here in this other organism can help with this process. So what we want to do is we want to take this gene from this other organism over here, who maybe encodes a special enzyme or something. Who knows? It doesn't matter for the sake of understanding why we're using PCR. But we want to take it and we want to use it to make many copies of this gene. So we can make many copies. And the reason we want to make many copies is it's kind of hard to take a gene from one organism and move it into another. The more copies we have, the easier it is to move that gene into another organism. So we're going to use PCR to make many copies, and that's going to allow us to take that gene and put it into the genome of our plant over here, and that's going to make our transgenic plant, or otherwise known as a genetically modified plant or genetically modified organism. So this PCR-based amplification of the gene, this is just one of many steps. Usually it's the first step that you would use to make a genetically modified organism. So we'll save all these other steps in here for our 300 level biology classes. Definitely molecular biology and our biotech classes here at ISU. Okay, so let's go on to talk about how we would use PCR to make many copies of this gene. Okay, so next, let's talk about what we need to do PCR. Well, first, we are going to want to take a cell here. We'll take our cell that we want to, that has the gene that we're interested in. So here's the nucleus and the chromosome, and we don't want to just work with this cell here. We want to work with thousands of these cells. And depending on if this organism is haploid or diploid, each of these cells is going to have either one copy or two copies of the gene we're interested in. And so let's imagine here we're working with many thousands of these cells, and these are closer to our screen here, so that's why they're so big compared to these ones, which are in the distance. But we're working with thousands of these cells, and what we want to do, well, we want to isolate the genomic DNA here. So we're going to isolate that using some standard protocols not too difficult to understand, but we want to learn about PCR. We don't want to waste the lecture talking about genomic DNA extraction methods. So we are just going to isolate this DNA, and we're going to have a tube here, and in our molecular biology and genetics laboratories, we like to work with these little tubes called MCT tubes or microcentrifuge tubes, and they hold about 1.5 milliliters. That's not that important. I just want to give you an idea of the scale, the size of these tubes. And these are very common. And in here we'll have our genomic DNA. Now this genomic DNA is going to have a lot of different genes, right? It's going to encode all the genes required by this organism to live. But it also has our gene, the gene we're interested in, the target gene, let's call it. And this genomic DNA is going to be the DNA template. So PCR requires a number of different components, and a DNA template is the first component we're going to need for PCR. So, okay, so next, nowadays things are made really simple for us. So we can order these relatively expensive kits called PCR kits, polymerase chain reaction kits. And inside this kit, we're going to have a number of other reagents we're going to need to do PCR. And again, these come in little MCT tubes. And one of the components of the kit is going to be a DNA polymerase. So we talked about DNA polymerases when we talked about DNA replication. And these DNA polymerases are very similar to those, and there's something special about this that we're going to talk about in a little bit. We're going to talk about what's special about this DNA polymerase. But okay, we get a tube with DNA polymerase. We'll get another tube with buffer, so PCR buffer. So the DNA polymerase needs a certain salt concentration in order to work efficiently, and that's going to be supplied by the PCR buffer. Some of the nicer kits even come with a special ultra pure supply of water. So ultra pure H2O. Okay, so also, what else are we going to need? We are going to need a solution of DNTPs. DNTPs. So what are these? We talked a little bit about this in chapter 10. So deoxyribonucleoside triphosphates. So we're going to be making copies of this gene. So of course we're going to need deoxyribonucleoside triphosphates to make the DNA. So this is just a collection of DATP, DGTP, DCTP, and DTTP. So the D is for deoxyribo because we're making DNA, not RNA. And at the 5' ends of these here, we are going to have 3 phosphates. And at the 3' ends, we're going to have an OH group. And remember the 5' carbon and a 3' carbon from our discussions of the structure of a nucleotide. Looks like I have to erase some things from an earlier YouTube video here, so let me do that. Okay. Okay, so where was I? Oh yeah, deoxyribonucleoside triphosphates, DNTPs, just a collection of the 4 nucleotides we're going to need to replicate the DNA. So we have a number of different components here. We need a DNA template, number 1. We need DNA polymerase. We need PCR buffer, which is number 3. We need water, ultra pure water here, which is number 4, and we need DNTPs, which is number 5. We're also going to need 2 more things. We are going to need what we call a forward primer and you should recognize the word primer, right, from our talk of replication, and reverse primer. But unlike in replication, these are not RNA primers. These are usually, well, as far as I know, they're always DNA primers. So 7 different things are needed. 7 different components are needed for a PCR reaction, is what we'll call it. Which is kind of redundant because then it's a polymerase chain reaction. But a lot of times we'll just say PCR reaction. Technically, we could probably drop off the reaction and just say for a PCR. But that sounds kind of funny, so I'll probably say PCR reaction. Okay, so now that we know about all the components we're going to need for a PCR reaction, we can go over the steps of a PCR reaction. So there are in general, there are 3 steps. Denaturation, annealing, and that is spelled incorrectly. Sorry about that. There's only one L in annealing. And C, extension. So 3 major steps. So let's go over these one at a time. Okay, so we mix everything in our PCR tube here, or even a test tube. We could even, let's just start by mixing everything in a test tube. So we have a test tube here, and we have all our components in here that we need to do PCR, all 7 of those components we talked about. Polymerase, etc. So we are going to try and copy that gene, that single gene within that genomic DNA. So one of the first things that we need to do is we need to denature the DNA, and this is the first step, right? So we talked about how DNA is denatured, so it can be denatured with chemicals, or we can denature it with heat. And remember, denaturation of DNA is just taking those 2 strands and separating them. And so we just need to break the hydrogen bonds, and we're going to do that with high temperature. So we could heat it up to 100 degrees, but in general, right about 95 degrees Celsius is thought to be hot enough to denature double-stranded DNA. So we're going to heat that to 95 degrees, and let's say, you know, there's lots of DNA in here, but let's say this is the DNA that holds the gene we want to amplify. Okay, and we are denaturing the double-stranded DNA, so now we have 2 single strands. And keep in mind that these have 5' ends and 3' ends, and it's a good idea to keep track of those ends when you're thinking about PCR. So 5', 3', 5', 3', 5'. Okay, so now that it's denatured, and how do we do this? So let's say we had everything in a test tube there. Here's our test tube again. And we just took that test tube and we put it in an incubator that was really hot. That was, put it in a little oven. So everything heated up to 95 degrees Celsius. Or we could use a water bath. We have these things called water baths, which are set at certain temperatures. And we could just take the test tube and dip it in that water bath and heat it up to 95 degrees Celsius. Of course, it would be capped. We wouldn't want anything to get in there. But we heat it to 95 degrees, and any double-stranded DNA in there is now denatured. And keep in mind, we have all our other components in there as well. So the next step is the annealing temperature. The annealing step of PCR. And we have to lower it to an annealing temperature. And in general, an annealing temperature of about 60 degrees is a good annealing temperature. And we're going to talk a little bit more about why we use this 60 degrees in a little bit. But what happens during the annealing step is those primers that we have are going to, anneal to either sides of the gene we want to amplify. So, and what do I mean by that? So let's take, okay, so here's one primer, and we designed this primer specifically. So let me talk a little bit about the primers first. So primers, I wanted to change that color. So primers in general are single-stranded DNA molecules of about 20 nucleotides long, and we can call these 20 mers. And sometimes they're could be as short as 18 nucleotides long, and those would be 18 mers. It can be as long as 50, sometimes even longer. But in general, we're going to use primers of about 20 nucleotides long, and we can call these 20 mers if we want. So these are single-stranded DNA, and what we do is we order them from companies, right? We can order this from a company, and they will make it for us, and we'll specify the sequence. And what we'll do is we'll make the reverse primer, so it will be complementary to this end of the gene right here. And I'm not going to draw all 20 nucleotides here, 20 or so nucleotides, but it will be complementary here. And this forward primer will be complementary right here on the other side of the gene. So you can see that the reverse primer is sticking to this top strand, and the forward primer is sticking to the bottom strand of the template. Now, okay, so the reason this is annealing is because we lowered the temperature. So we're no longer at a temperature that causes the double-stranded DNA to denature. We're at a temperature that allows the primer to anneal to the complementary sites on the genomic DNA. Okay, so far we, and I should also say that the template strands could come back together, right? But we put so much primer in there that the primer is more likely to find the complementary site than is the long piece of DNA here. So that's one reason why the primers anneal preferentially to the complementary DNA before the complementary strands of the templates. Okay, so that's the annealing step of PCR. So now let's talk about the extension step. What I want to do here, I want to keep this figure right here. So let's see if I can erase some things. I'm going to cut that, I'm going to cut all that out, and we'll keep our tube here. So what do we do? How do we lower that? I'll step back a bit. How do we move down to 60 degrees? Well, what we did was we could have just taken this test tube and moved this to a different incubator or a different water bath set at 60 degrees Celsius. Okay, so that's all we need to do. We need to change the temperature of the reaction, and that is going to go from the denaturation step to the annealing step. And because all the components are here in the solution, this is going to happen automatically. Okay, so now we want to extend these primers. And, well, we aren't going to do it. We want it to be extended. The polymerase is going to do the extension. So what we need to do is raise this to a temperature that is optimal for the polymerase. So we're going to raise this to about 72 degrees Celsius. And this is going to be called the extension step. And this is the extension temperature. So this is the extension step of PCR. And what happens is that polymerase now, its job is to polymerize the DNA. So it's going to take those DNTPs and it's going to add them to the 3' ends of the primers. And it's going to extend each strand of the template. Okay, so what we're going to do, we'll talk a little bit more about this in a moment, but we're going to give the extension step about 1 minute per 1,000 base pairs of the length of the target. We said our gene was about 3 kb or 3,000 base pairs, so we would run this extension step for about 3 minutes. Okay, and then what will we do? So we run it for about 3 minutes, and what have we done? Well, we've taken, if we started with just one copy here, if we started with just one copy of the gene, at the end of the extension step, we now have 2 copies, right? So that's not that much more DNA. That's not, we haven't amplified it that much. We've just doubled the number of copies. So we want to keep doing this, and what we're going to do now is we are going to cycle these steps. We're going to go from the extension step, we're going to take that test tube at 72 degrees and we're going to raise it back up to 95 degrees. And what is that going to do? Well, that's going to make those 2 new copies of DNA we just made, of those 2 new copies of the gene, it's going to denature them, and then we can repeat the process. So let's say we now have 2 copies of the gene here. We're going to denature those by cycling back up to the 95-degree temperature. So we're just taking our test tube, we're putting it at 95 degrees, the DNA denatures. Remember, we had 2 copies after that first round, after that first cycle. So we raise the temperature to 95 degrees Celsius, and then we take that test tube and we put it at 60 degrees Celsius, and that's going to allow our primers to bind. So I stopped labeling the ends here, but we have 5 prime, 3 prime, 3 prime, 5 prime. Remember, the primers have 5 prime and 3 prime ends, and they have to match up with the templates. So the primers are annealing at 60 degrees Celsius. My hydrogen bonds are kind of messy here, but I think you know what's going on there. And then we raise the temperature, take that test tube and put it now at 72 degrees Celsius, and the polymerase binds here and extends these primers, so we make more copies of our DNA. Okay, so we started with, let's say we only started with 1 copy of this gene. Now, after 2 cycles of these temperatures here, we now have how many copies? My drawing is kind of messy, but I think we can make out here that, you know, if you're keeping track, you probably already realized, but I can also circle these. We now have 4 copies. So if we wanted to calculate, you know, how many copies we're going to make with each round of replication, we have a certain formula for that. Let's see how this formula works. So the formula is number of copies equals 2 to the n, where n is the number of cycles. So let's say we didn't cycle at all. Let's say we had 0 cycles. We never even ran the reaction. How many numbers would we have? So here's 0 cycles here. We'll have a table here. 0 cycles, number of copies. So 2 to the 0 equals 1. Okay, 1 copy, that makes sense. If we had just 1 cycle, well, that would be 2 to the first power here, and that would be, how many? That would be 2, right? Okay, that makes sense. We started with one round of PCR. We have 2 copies. How about 2 cycles? 2 to the second here, we have 4 copies. What if we raise this to the 25th power? We do 25 cycles. Well, you can see we're going to have millions of copies. We could even go higher, you know, 2 to the 30, 2 to the 35th power here, and maybe that goes up to billions. I'm not sure. So we can't do this indefinitely, right? Because we are going to run out of primers, and eventually that polymerase isn't going to work anymore, because although the type of polymerase we are using can survive 95 degrees Celsius, it can't survive it forever. So eventually it's going to break down. So we're going to run out of the reagents, and we can't really go much higher than 25 or 35 cycles to see that much difference in the amount of product or the amount of copies of the gene we're interested in. So just in 25 rounds of PCR, we can go from one copy of the gene we're interested in to millions of copies of that gene. And keep in mind, every other gene that was in that genomic DNA we started with, it wasn't amplified. So essentially we have a tube of the gene we are interested in now. And that's going to make these downstream processes of putting that gene in another organism much easier. So let's talk about some details of the PCR process. One of the things we need to do if we're going to try and have a successful PCR reaction is we need to design good primers. And there are a couple of considerations we need to take into account when we are designing primers. We are only going to be concerned with the melting temperature of the primers. We'll worry about the other things when we get to our 300 level biology courses. So TM, so this is melting temperature, and you probably remember from an earlier part of the course where the melting temperature of a solution of DNA is the temperature at which half of the DNA is single-stranded and half of the DNA is double-stranded. And the temperature depends on usually depends on a number of things, but most importantly GC content. So this is what we talked about in the past, right? And that DNA solution can be all of the DNA in a genome of an organism, or it can be one gene. Many copies of one gene. When we're talking about our primers, so when we're talking about the TM of a primer, well, let's imagine that we have a tube of just our primer, and now we're going to add to that tube, say, an equal amount of the the complementary sequence. So another primer that is perfectly complementary, so that it could form a nice short double-stranded DNA molecule here. So the TM of our primer is going to be the temperature at which half of the DNA in our solution of primer and its complement are single-stranded, half single-stranded, and half double-stranded. Okay, so that's going to be the TM of our primer. And keep in mind there are going to be two primers that we need to use, and we need to consider the TM of both primers in our PCR reaction. Okay, now why do we care about primer TM? So let's go back to that transition between the denaturation step and the annealing step of our PCR reaction. So let me diagram just one of the strands of the DNA template here. I might just I'm going to copy this and save it, so next time I can just paste it into the screen here, and so I don't have to diagram this each time. But hey, at least you're getting some time to think about what we're talking about. Okay, so here is one of the strands of our template, and so we have lowered the temperature. We took our test tube and we put it in an incubator at, let's say, 60 degrees Celsius. So we're going to put it in an incubator at 60 degrees Celsius for our annealing step. And let's say for our forward primer here, let's say the TM of our forward primer is 65 degrees Celsius. Okay, now that primer, we want it to stick right about here. Why? Because we designed the primer specifically so it's complementary to this site right here on the template. Now we have an annealing temperature of 60 degrees Celsius. and our primer TM is 65 degrees Celsius. So what if our annealing temperature was not 60 degrees? What if it was, 70 degrees? What if we took that test tube and we went from 95 degrees, and instead of lowering it to 60 degrees, we put it in a 70 degrees Celsius incubator? How much of our primer here would we expect to bind to this site right here, given that the TM is 65 degrees Celsius? So would more than half of the primer bind, or less than half? Right? So we would expect, I don't want to confuse you all and confuse myself too much, but at a high temperature of 70 degrees Celsius, we would expect less than half of all the primer to be able to bind to there, or the primer would bind, let's say, less efficiently. Because we know the TM 65 and 65 degrees Celsius is the temperature at which half the primer is in the double-stranded form and half is not. So at 70 degrees Celsius, we would expect, less than half to be in the double-stranded form. So let's go with less than half. And how about at 60 degrees Celsius? Well, that's a good temperature, and that promotes annealing of the primer. So more of the primer is more likely to be in the double-stranded form. So at 60 degrees Celsius, we have, let's say, more than half, is more likely to be in the double-stranded form. So that's how I like to think of these annealing temperatures and why we choose annealing temperatures the way we do. So we know we have the melting temperature. We know below the melting temperature of the primer, more of the primer is going to be in a double-stranded form, and above the melting temperature, less of the primer is going to be in the double-stranded form. So you might be wondering, why don't we just lower it to 30 degrees Celsius or room temperature? Maybe we don't even need an incubator. Maybe we can just use our hand, and that would be about 37 degrees Celsius. So, and there's a good reason why we don't do that. And I said I was going to copy this and paste it, but I forgot. So let's see if I can copy this strand right here. It's going to be a little messy, but okay, that's okay. So here's our template again, and let's say we lowered, instead of the 60 degrees Celsius, we lowered the temperature to, 30 degrees Celsius. Okay, now we want the primer to stick right here. This is our primer, this is our target binding site. If we go too low, however, well, the primer can start to stick to other parts of the target, or other places in the genome, even places where it's not perfectly complementary. So it might be partially complementary. Say these first 3 bases right here are complementary, but let's say these guys over here aren't, so we're not forming hydrogen bonds here, but then let's say these couple are. So what can happen is you can get what we call mispriming. So the primer is actually annealing to a part of the genome that is not complementary. And it can do that at these very low temperatures, because you don't need all the hydrogen bonds to form here in order to keep these guys stuck together at these low temperatures. There's less energy in the system, and we're not going to go into the thermodynamics of this process. So ideally, what we want to do is find that perfect annealing temperature to promote binding of our primer to the sequence that is perfectly complementary to it. So we don't want to go too far below the melting temperature of the primer when we're choosing the annealing temperature of our PCR reaction. So far we have talked about using a denaturation temperature of 95 degrees, an annealing temperature of 60 degrees, and an extension temperature of 72 degrees. So this is our annealing, and we actually change this based on the TM of both of our primers. So we take the lowest TM. We have 2 primers, whichever one has the lowest TM, we take that, let's say the lowest TM was 65 degrees Celsius, we just subtract 5 degrees Celsius from there. And then we have our annealing temperature of 60 degrees. So if we have two primers, primer 1, primer 2, primer 1 has a melting temperature of 64 degrees, primer 2 has a melting temperature of 62 degrees Celsius, then what would we set the annealing temperature to be? Well, we take the lowest one and we subtract 5 degrees. So that would be 57 degrees Celsius. Okay, so we just modify the temperatures in our PCR reaction. So now we have 95, 57, and 72. So how can we figure out the melting temperature of our primers? We have fancy programs, software that uses complicated equations to figure this out in our laboratories. But you know what works just as well, especially for primers that are about 18 to 24 nucleotides long. So for primers within this range, we have a shortcut that we can use to allow us to approximate the TM of a primer by hand. So we just take the sequence of our primer, we add up the number of A's and T's, and we multiply that number by 2. And then to that, we take the primer sequence, we add up the number of G's and C's in that primer, and we multiply that by 4. We add those together, and we get the TM of the primer, or the approximate TM of the primer. And this works fairly well for primers that are about 18 to 24 nucleotides long. Anything longer than that, and you're going to need to find a program, and that will use some complicated formula to approximate the TM. Now, okay, so let's say, let me give you an example here. So here's a forward primer, and let's use, let me see, GG, A, C, C, T, A, G, C. How many we have here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And I'm just going to copy that again. So that should be 20, right? Well, I could just count these up. So let's say this is our primer sequence, and this is complementary to the end of the piece of DNA that we're trying to amplify by PCR. So let's count the G's and C's. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So we have 11 G's and C's. Let's count the A's and T's. 1, 2, 3, 4, 5, 6, 7, 8. So 8 A's and T's. This is a relatively short primer. So I might want to make it a little bit longer, but let's see what the TM is. I'm going to multiply this by 4. I'm going to multiply this by 2. So we're going to have 44 here. We're going to have 16 here. This is going to equal, when we add these together, this is going to equal 60. So we're going to want an annealing temperature of 55 degrees Celsius, assuming that, you know, this is the lowest TM. This primer TM is lower than the TM of the reverse primer. So pretty easy, right? Pretty easy to choose the annealing temperature. Okay, so now let's take a little look at, or let's talk a little bit about the DNA polymerase for PCR. There are some special things about this DNA polymerase which makes PCR possible. So one of the things you may have noticed is that we are using really warm temperatures during PCR, right? 95 degrees Celsius is near boiling, right? What do you think would happen if most of our proteins were exposed to, you know, boiling temperatures like this? They would probably not function. Even 72 degrees Celsius, this is the optimum temperature for this polymerase we're using. This is really hot. Our bodies are about 37 degrees Celsius. For a long time, scientists didn't think life could survive above 55 degrees Celsius. So no life above 55 degrees Celsius. Even the annealing temperature that, you know, we typically use in my lab, but even this annealing temperature right here of 60 degrees is above this, you know, 55 95 degrees theoretical limit that scientists used to think was the limit, maximum temperature life could survive at. But there are organisms called thermophiles. And these thermophiles thrive in extreme temperatures. And there is an organism called Thermus aquaticus, and this organism was found living near hot springs in Yellowstone Park, and it thrives at 72 degrees Celsius and can survive up to 80 degrees Celsius. So this was a huge finding. You can probably imagine that an organism that has evolved to live near these hydrothermal vents in hot springs, all of its proteins are adapted to function at these high temperatures. And the DNA polymerase, or one of the DNA polymerases from this organism, we're going to call this DNA polymerase TAC DNA polymerase, for Thermus aquaticus DNA polymerase, TAC DNA polymerase. So this polymerase can survive, at 95 degrees Celsius. It's not going to function at 95 degrees Celsius, but it doesn't break down rapidly. It can survive, I think, up to like 20 or 30 minutes at 95 degrees Celsius, and it can still function. So the discovery of this polymerase allowed PCR to happen because it needs to work at those high temperatures in order to have the primers annealing at the right position, at the right spots on the target during the annealing step, in order to denature the DNA to allow multiple cycles to occur. So the polymerase needs to be able to survive at this high temperature. So scientists working with thermophiles discover these organisms that could live in extreme temperatures. The organisms have proteins that function well at these high temperatures and can survive near boiling temperatures and still function. And these are the enzymes used in these techniques like PCR, which require high temperatures. So let's sum up what we've talked about during this little video on PCR. Okay, if we're going to perform a PCR reaction, in general, we're going to have a number of different steps. We talked about the key steps of a PCR cycle: denaturation, annealing, and extension. But let's just organize this a little bit. So in the first step, we are going to denature the DNA. We've put everything in our test tube, and we're going to denature it. And scientists have already worked out the perfect, or the optimal length of time for all of these steps. And so we're going to denature at about 95 degrees Celsius for 30 seconds. Okay, and in step 2, now we're going to do something weird here. We're going to denature again. So we've already denatured once, but we're going to have a step 2 denaturation step at 95 degrees Celsius, and now we're only going to do it 5 more seconds. Okay, 5 more seconds. So step 3, this is our annealing step. So we're going to lower to the annealing temperature. And let's say our TM, the lowest TM of one of our primers was 65 degrees. So we'll set the annealing for 60 degrees Celsius. And we're going to run this for 15 seconds. So only 15 seconds are needed for that primer to find the right spot on the template and bind. Step 4, this is our extension temperature, and we are going to run that at 72 degrees Celsius, the perfect temperature for the best function of our DNA polymerase. And we are going to do this for 3 minutes, right? Because our target is 3,000 base pairs. So we want about 1 minute per 1,000 base pairs of target. So we're going to do 3 minutes there. Now step 5, this is going to be our cycling step. So what we can do here is we can sit here by these tubes and we can move it back and forth for how many times? The more times we do it, the better. We talked about a theoretical maximum of about 35 times is about, wasting our time after that. But we are not going to go back to number 1. We are going to go to step 2. So 25 times we're going to cycle between steps 2, 3, and 4. So the reason we do that is we want to minimize the amount of time our polymerase spends at 95 degrees Celsius. So we do an initial denaturation just to make sure all that template DNA is denatured of 30 seconds, a long one, and then we've learned that each subsequent denaturation step only has to be relatively short for PCR to work just fine. So we're going to cycle back to number 2 25 times. And then, after we do that, step 6, we are just going to, put our reaction in the freezer. So we've got, if it works, and it usually works, especially if you have a little bit of experience with this, millions of copies of our target DNA. Now we can move on to the next steps to putting this in an organism if we're interested in making a transgenic organism. If we're sequencing parts of a gene that we think might have mutations, we can sequence that DNA now. Many different things we can do with that DNA. So next I think I just want to touch on one more thing. Okay, yes. So far we've talked about doing all of this in a test tube, and what we can do, okay, we can have just a test tube right here, right? And we have a big reaction here, I don't know, 1 milliliter maybe, and we generally don't need to do large volumes like this for our PCR reactions. What we usually use, these little PCR tubes. These are very common. And these hold about 200 microliters or 0.2 milliliters. And we do these little miniature reactions, maybe only 0.05 milliliters. So all of our components are put in here. And one thing we haven't talked about is the concentrations of all of the different components, the amount of primer, the amount of genomic DNA for the template, the amount of enzyme and DNTPs. There are certain concentrations that we should use for the PCR reaction to work. We don't need to worry about those. But we normally do our reactions in these little tubes. Scientists all over the world do their reactions in these little tubes, and they don't sit by the tubes and move them from incubator to incubator. What they use is a thermal cycler. So thermal cycler, and you can imagine what a thermal cycler does. It just changes the cycles of the temperature of a reaction. So I can copy and paste an image of a thermal cycler right here. So we mix up our reagents for the PCR reaction in this little tube, and then we put those little tubes, into the little, right here we can see we have 96 of these little holders right here for the tubes. And what the thermal cycler will do, it will rapidly change the temperature surrounding the tube. So it leads to rapid changes in temperature. So we can cycle between 95 and 60 and 72 very quickly. And we don't need to sit next to the tube and move it back from oven to oven or water bath to incubator to whatever. We just put those tubes in there and we program all the different steps into here. So the thermal cycler knows what to do and what needs to know, or knows what temperature to expose the tube to for how much time for each step. So it's very easy, and when it's done, it will hold the tube at 4 degrees Celsius or 12 degrees Celsius or at a cold temperature. So you can set up your PCR reactions and go home and come in the next day and analyze the products. Okay, so relatively easy. And PCR reactions can be kind of fun, and I hope you do take biotech. If you're not working in a genetics lab or a molecular biology lab here at ISU, then I do hope you take biotech, our 300 level laboratory classes, where you will do a lot of PCR. So we have a couple other things. I hope you check out the lecture notes on this lecture, and we have 3 problems 20.12, 20.20, and 20.21 from our textbook that you should read, and after watching this video you should be able to answer those questions. And you can also read chapter 20 section 3, which is our textbook's introduction to the polymerase chain reaction. If you have any questions, just come see me at office hours or make an appointment. Okay, well, I'll see you in class on Tuesday.